I'm Dr. Emily Carter, and today we're talking about something that lies at the heart of modern cytogenetics: structural variants, those large scale changes in our genome that can be both creative and catastrophic.
And I'm Daniel Ross. Great to be here, Emily. It's funny, when people think about genetic variation, they usually picture single-point mutations, those tiny changes in a single letter of DNA. But the real architects of genomic diversity are often the big players: deletions, duplications, inversions, and translocations, collectively called structural variants.
Exactly. These variants can rearrange chunks of DNA that range from a few hundred base pairs to millions. They don't just tweak the genome, they rewrite it. What's fascinating is that the very same mechanisms that allow our species to adapt and evolve can also make us vulnerable to disease.
Right, that duality is what makes them so interesting. On one hand, structural variants drive gene innovation, diversity, and adaptation. On the other, they're implicated in everything from developmental syndromes to cancer. It's the genome's double-edged sword.
Let's start with the basics. Daniel, for listeners who aren't deep into genetics, what creates these structural variants?
Great question. The genome is filled with repeated sequences, stretches of DNA that look almost identical in different places. These repeats can confuse the cell's repair machinery. During DNA replication or recombination, the system can misalign these similar sequences, leading to mistakes. Sometimes entire segments are duplicated or deleted. We call one of the main processes non-allelic homologous recombination, or NAHR.
So it's like the genome's spell check system skipping a line because two paragraphs look the same.
Exactly. That analogy works perfectly. Another major mechanism involves replication errors. When a DNA strand stalls during replication, the polymerase may jump to a different template and then back again. We call this fork stalling and template switching. It can create very complex rearrangements.
So these are not random events. The architecture of the genome itself, all those repetitive elements, actually predisposes certain regions to instability.
Absolutely. Some areas are what we call genomic hotspots. They're fragile but also evolutionarily active. Many of our gene families, olfactory receptors, immune genes, detoxification enzymes, have expanded through repeated duplications in these same regions.
Which brings us to the evolutionary side of the story. Structural variants don't just cause problems, they've been engines of innovation throughout evolution.
Definitely. Think of gene duplication as evolution's favorite trick. When a gene gets copied, one copy can keep doing the essential job, while the other is free to mutate and experiment. Over time, that can lead to new functions, new enzymes, receptors, even developmental pathways.
A famous example is the AMY1 gene, which codes for salivary amylase. Populations with high starch diets have more copies of AMY1 than those with low starch diets. That's structural variation, shaping metabolism and adaptation in real time.
And yet that same flexibility comes at a cost. The very mechanisms that allow duplication can also delete critical genes or disrupt chromatin structure. For instance, if a duplication changes the boundaries of what we call topologically associating domains or TADs, the result can be disastrous.
Let's pause there. TADs are such an important concept. These are 3D domains in the nucleus, right? Regions of DNA that loop together and interact internally, like neighborhoods of genes and their enhancers.
Exactly. Each TAD is a regulatory unit. It ensures that specific enhancers, the on switches of genes, talk only to their correct target genes. When a structural variant breaks a TAD boundary, enhancers can get rewired. They may start activating the wrong genes in the wrong tissues.
There's a striking study by Lupianas and colleagues where they showed that rearrangements near the EPH4 gene can cause limb malformations in humans and mice. The rearrangement moves enhancers from one domain to another, so they start activating genes that should be silent during limb development.
Yes, that's the perfect example of enhancer hijacking. The DNA sequence is all there, no genes are missing, but the spatial architecture is wrong. The genome's 3D structure has been scrambled.
It's mind-blowing, isn't it? The genome isn't just a linear string of code. It's folded, looped, and compartmentalized, and those folds are essential to how it works.
And that's why cytogenetics today isn't just about chromosomes under the microscope anymore. We've moved into integrative cytogenomics, combining molecular sequencing, chromatin conformation mapping, and classical cytogenetics imaging to understand the full picture.
So, technologies like Hi-C and 4C-seq let us see these long-range interactions, while sequencing gives us the precise breakpoints of structural variants. When you merge those data sets, you can really predict the functional outcomes, whether a CNV might change dosage or if a rearrangement might disrupt a TAD.
Exactly. It's a synthesis of methods, from karyotyping in fish to next-generation sequencing and 3D genome mapping. That's the future of medical genetics.
Let's bring the conversation back to disease. CNVs are now known to play major roles in neurodevelopmental disorders, right?
Yes, deletions or duplications at certain hotspots like 16p 11.2 or 22q 11.2 are linked to autism, schizophrenia, and developmental delay. What's fascinating is that deletions and duplications of the same region can produce mirror phenotypes. For example, microcephaly versus macrocephaly, or underweight versus obesity. That symmetry highlights how sensitive development is to gene dosage.
So, the same mechanisms driving gene expansion and adaptation can also underlie severe clinical syndromes. Evolution and pathology share the same molecular grammar.
That's beautifully said. We often think of evolution as constructive and disease as destructive, but they're two sides of the same coin. Genomic instability fuels both.
It's almost poetic, the genome experimenting with itself, sometimes producing innovation, sometimes error.
Exactly. And evolution doesn't distinguish between good and bad mutations. It just experiments endlessly. Structural variants are part of that experiment.
I think one of the key takeaways for students and researchers is that we can't view structural variation only as pathology. These variants reveal how dynamic our genome truly is, constantly balancing flexibility and stability.
Well put. In fact, some CNVs persist in populations because they're conditionally advantageous. The UGTB17 deletion, for instance, influences steroid metabolism. It can reduce the risk of certain conditions but increase others, like osteoporosis. That's genomic trade-off in action.
So, if we zoom out, structural variants are both evolutionary experiments and medical challenges. Understanding them helps us trace human history and treat human disease.
Precisely. And as sequencing technologies become even more precise, we'll start seeing SVs not as exceptions, but as a fundamental layer of human variation.
Daniel, before we close, what excites you most about where this field is heading?
Honestly, the integration of 3D genome mapping with clinical diagnostics. We're getting to a point where a cytogenetic analysis won't just tell you something's missing or duplicated, but will predict how that structural change affects gene regulation in 3D space. That's the bridge between structure and function.
I couldn't agree more. It's an incredible time to be studying cytogenetics, a field that's evolving as fast as the genome itself. A huge thank you to Dr. Daniel Ross for joining me today. If you enjoyed this episode, share it with your fellow genetics enthusiasts. Until next time, remember, the genome is not static. It's alive, dynamic, and always rewriting its own story.
Thanks for having me, Emily.